In addition to the complexity of inheritance, or the simplicity of Mendelian inheritance impacted by other factors, nature and the environment can often have an impact on phenotype. So, the norm of reaction is going to be the phenotypic range of a genotype that is influenced by the environment. So, for example, in hydrangea flowers, you can have a genotype range from blue-violet to pink, but it all depends on soil acidity. So on the left-hand side, you'll see the pink, and on the right-hand side, you'll see the blue hydrangeas. The norms of reactions are broadest for polygenic characters, so characters that are inherited by more than one gene, or that are influenced by more than one gene. And these are called multifactorial, because both genetic and environmental factors are going to influence the phenotype. An organism's phenotype is going to include its physical appearance, as we know, but it also includes the internal anatomy and physiology, and also sometimes the behavior of an organism. It's going to reflect the overall genotype and the unique environmental history of that particular organism. So, many human traits are going to follow Mendelian patterns of inheritance, so therefore we're not particularly good subjects for genetic research because our generation time is too long. It takes many years, sometimes decades, to produce offspring. Parents produce relatively few offspring, and it's not acceptable socially, or some would say it's even unethical, to do breeding experiments on humans. But basic Mendelian genetics, therefore, endures as the foundation of human genetics. There are many genetic disorders that can be inherited in a recessive manner in humans, and these range from mild disorders to disorders that can be life-threatening. So, remember that recessively inherited disorders are only going to show up in individuals that are homozygous for the allele. So they have to have both recessive alleles in order to show the disorder in their phenotype. So carriers are going to be heterozygous individuals. They have the recessive allele, but phenotypically they appear normal. And most individuals that have recessive disorders are born to parents that are both carriers. So albinism is a recessive condition characterized by lack of pigmentation in the hair or the skin. So as you can see, if two parents appear normal, they can have offspring. They have a 25% chance or a 1 in 4 chance of having albino offspring by that individual inheriting both recessive alleles from each parent. If recessive allele is rare, then the chance of having two carriers meeting and mating is very low. There are, however, consanguineous matings, which we would refer to as incestuous matings, and those are going to increase the chance of mating between two carriers of the same allele, same rare allele, and so most societies and cultures have laws or they frown upon marriages between close relatives because those recessive alleles that are carried within families tend to show up more often when close relatives mate. Cystic fibrosis is an inherited genetic disease, and it is the most common lethal genetic disease in the U.S. It strikes 1 out of every 2,500 people of European descent. Nora, I need quiet, please, honey. The cystic fibrosis allele results in a defective or absent chloride transport channels in the plasma membranes, so chloride ions build up outside the cell, and sometimes this can lead to symptoms like mucus buildup in some of the internal organs, and also an inability to absorb some nutrients in the small intestine. Sickle cell disease is another genetic disorder that impacts humans, and this one has evolutionary implications because about 1 out of every 400 African Americans is affected with sickle cell disease. It's commonly caused by the substitution of a single amino acid in the hemoglobin protein, which of course you would find in red blood cells, and it results in an abnormally shaped hemoglobin molecule, which is sickle shaped. And some of the symptoms of this disease include physical weakness, pain, organ damage, or paralysis. However, heterozygotes, individuals that have a normal allele and a sickle cell allele, are usually healthy, but sometimes they suffer some symptoms. And 1 out of every 10 African Americans has this sickle cell trait, which is a very unusually high frequency. Especially if there are going to be detrimental effects. Nora, please sit down. Heterozygotes are less susceptible to the parasite Plasmodium, which causes malaria. However, so there is an advantage to being heterozygous, because Plasmodium needs a normally shaped red blood cell in order to complete its life cycle, which results in malaria in humans. Some human disorders are actually caused by dominant alleles. Achondroplasia is a form of dwarfism. Also, Huntington's disease is caused by a dominant allele. So here is an example of how that would pass on from generation to generation. Huntington's disease is a degenerative disease of the nervous... Oh my goodness, I cannot speak today. I'm sorry. Degenerative disease of the nervous system. And typically, until the individual is about 35 or 40, there are no obvious physical effects. But once the nervous system starts to deteriorate, the condition is irreversible and fatal. There are other multifactorial disorders, meaning that not only do genetics play a role, but the environment also plays a role, such as... Nora, hold on, please. Nora, that's awesome that you have an aunt. I apologize. There are many diseases, such as heart disease, diabetes, alcoholism, mental illness, and cancer, that are going to have both genetic and environmental components to them. And it's not very well understood how the genetic contribution plays a role in most multifactorial diseases. However, with the Human Genome Project and some of the other research that's been going on lately, it's becoming more and more clear every day. And so scientists will possibly soon have cures to many diseases, or soon have preventative measures to take against many of these multifactorial disorders that have been previously ill understood. There are individuals who are trained in counseling counseling people on their genetics and the results of their genetic testing, called genetic counselors. And genetic counselors can give information to parents in particular about the probability of their offspring having a particular genetic disorder or disease. So this counseling is typically based on Mendelian genetics and probability rules, although there are some blood tests now that can give a risk percentage or a risk assessment of more complex types of multifactorial diseases. So these genetic counselors use family history, and they help their couples determine the odds that children will have certain genetic disorders. And they're predicted on the most accurate information at the time, but when an individual presents more information about their family history, of course, that could change the probability. So the probabilities that are predicted are only as accurate as the information that is provided to the genetic counselor. There are a growing number of diseases that have tests that are able to identify carriers, and then the odds are able to be defined more accurately. So here is a picture of a lab, and they're doing some form of a test. I actually used to do, I used to collect the samples and send out blood for some of these tests. It's quite interesting. When you read the reports there, they are rarely as well defined as any parent would like them to be when trying to determine whether or not their offspring will have some type of genetic disorder. So, which brings us to fetal testing. So there are different ways to test a fetus for various genetic disorders, and one of them is amniocentesis. And in amniocentesis, a very large needle is inserted into the abdomen of the pregnant female, and the liquid, the amniotic fluid that bathes the fetus, is removed and then tested for genetic disorders. There's also a technique called chorionic villus sampling, or CVS, and that removes a sample of the placenta, and then the placenta gets tested. Then there are, of course, other things such as ultrasound and fetoscopy, and those allow fetal health to be assessed visually in utero, and there's typically no harm to either the fetus or to the mother. So A here shows you a picture of an ultrasound and amniocentesis. So the ultrasound is used as a guide as to where to draw the amniotic fluid from so that the fetus isn't harmed by the procedure. And then that fluid will be placed in a centrifuge, and the parts will be separated out by their density, and then they can analyze a karyotype from those types of cells. And they do the same, essentially. The result is a karyotype for CVS as well. It's very common these days for newborns to be screened for genetic disorders. Prenatal screening is usually optional for parents, but a lot of newborn screening is mandated by law in various states, especially in New York State. So one of the things, for example, that they would screen for would be lead poisoning. Okay, so here we have basically a couple of figures that are going to help you organize all of the thoughts and all of the information that we have in this lecture here. So, if I recall correctly, epistasis is one of the words that you will not be expected to remember. If you look back at your curriculum framework, it will help you determine whether or not I'm telling you the truth. And polygenic inheritance is, however, definitely something that you need to know for your Regents exam. And I would argue that genetics is one of the most important units anyway, because the more we learn about genetics, the more you can learn about yourself, and the more you can learn about your offspring, and the more you can understand how your actions and the foods that you eat and the exercise that you do, how that's going to impact your offspring now. What you do now will influence your offspring that you might potentially have in the future. Okay, so that is going to sum up, to end currently our discussion of Mendelian genetics, and I apologize for the little Nora interruptions.